Hi everyone. Today we are going to see about Meselson and Stahl's experiment. This experiment was conducted by two famous scientists, Matthew Meselson and Francis Stahl. Introduction: The Meselson-Stahl experiment by Matthew Meselson and Francis Stahl in 1958, which supported Watson and Crick's hypothesis that DNA replication was semi-conservative. This experiment provided strong evidence that DNA replicates in a semi-conservative manner, where each strand of the original double-stranded DNA molecule serves as a template for the synthesis of new strand.
Hypothesis. Three hypotheses had been previously proposed for this method of replication of DNA. One is semi-conservative hypothesis, then the second one is conservative hypothesis, and the third one is dispersive hypothesis. Semi-conservative hypothesis. This method was proposed by Watson and Crick. The two strands of a DNA molecule separate during replication. Each strand then acts as a template for hypothesis of a new strand. Conservative hypothesis. The conservative hypothesis proposed that the entire DNA molecule acted as a template for the hypothesis of an entirely new one. According to this, histone proteins bind to the DNA, revolving the strand and exposing the nucleotide bases for hydrogen bond. Dispersive hypothesis. The dispersive hypothesis is exemplified by a model proposed by Max Delbruck, which attempts to solve the problem of unwinding the two strands of the double helix by a mechanism that breaks the DNA backbone every 10 nucleotides or so, untwist the molecule and attach the old strand to the end of the newly synthesized one. This would synthesize the DNA in short pieces alternating from one strand to the other. Each of these three models makes a different prediction about the distribution of the old DNA in molecules formed after replication. In this conservative hypothesis, after replication, one molecule is the entirely conserved old molecule and the other is all newly synthesized DNA. The semi-conservative hypothesis predicts that each molecule after replication will contain one old and one new strand. The dispersive model predicts that each new molecule will contain a mixture of old and new DNA. Experimental procedures. Nitrogen is a major constituent of DNA. 14N is by far the most abundant isotope of nitrogen, but DNA with the heavier 15N isotope is also functional. E. coli was grown for several generations in the medium containing NH4Cl with 15N. Then, when DNA is extracted from these cells and centrifugated on a salt density gradient, the DNA separates out at the point at which its density equals that of the salt solution. The DNA of the cells grown in 15N medium had a higher density than cells grown in normal 14N medium. DNA was extracted periodically, and when compared to pure 14N DNA and 15N DNA, after one replication, the DNA was found to intermediate density. Since conservative replication would result in equal amounts of DNA of the higher and lower densities, conservative replication was excluded. However, this result was consistent with both semi-conservative and dispersive replication. Semi-conservative replication would result in double-stranded DNA with one strand of 15N DNA and one of 14N DNA, while dispersive replication would result in double-stranded DNA with both strands having mixtures of 14N and 15N DNA, either of which would have appeared as DNA of an intermediate density. Finally, result. DNA from cells after two replications had been completed was found to consist of equal amounts of DNA with two different densities, one corresponding to the intermediate density of DNA of cells grown for only one division in 14N medium, the other corresponding to the DNA from cells grown exclusively in 14N medium. This was inconsistent with dispersive replication, which would have resulted in a single density lower than the intermediate density. of the one generation cells, but still higher than cells grown only in 14N DNA medium, as the original 15N DNA would have been split evenly among all DNA strands. The result was consistent with the semi conservative replication hypothesis. Thank you.